Hello, welcome back. Here we are moving on to the last region of the brain, which is the cerebrum. But we are going to talk about another functional region of the brain called the limbic system. It's not really a structure like the pons is a structure, the cerebellum is a structure. It uses the structures of existing parts of the brain, part of the diencephalon and the cerebrum, to do particular functions. So a lot of times it's called a functional region of the brain, as opposed to an actual anatomical region of the brain. So taking a look at these diagrams, we can see we're taking a slice down the longitudinal fissure, separating the hemispheres, looking at one hemisphere. So just to orient us as to where we are, so remember this was our thalamus, this was our hypothalamus, and this is our epithalamus. So this whole region was the diencephalon. So there's going to be some integration of the limbic system with structures within the diencephalon, primarily the hypothalamus. And there is that anterior group of the thalamic nuclei that communicates with the limbic system as well. And then we also are seeing kind of this green highlighted area here, which is part of the cerebrum. So the limbic system kind of straddles both diencephalon and cerebrum when it comes to the structures that are being utilized for these particular functions. Look here. In those functions, which are listed here in the notes, we have the limbic system establishes emotional states, linking the conscious intellectual functions of the cerebral cortex with the unconscious autonomic functions of the brainstem, and it helps to facilitate memory, storage, and retrieval. So emotional states, linking conscious and unconscious functions, and memory, storage, and retrieval. So those are the major roles that the limbic system plays in brain function and processing. So some of the key areas, I guess, since it's not really a structural area, we have what's called the limbic lobe right here. So the limbic lobe includes a few of the gyri or gyrus, which are the wrinkles of the cerebrum. So you have the cingulate gyrus, the perihippocampal gyrus, and the dentate gyrus. They're just these regions, these wrinkles, ridges on the cerebral cortex that are involved in some of these functions. We also have a few nuclei. We have the amygdaloid body, or sometimes just called the amygdala, and we have the hippocampus, which sits within the dentate gyrus, that wrinkle of the cerebrum. Now, the amygdaloid body plays a role in some of your subconscious control of things like your regulation of your heart rate. It helps to connect to your fight or flight, so that's our sympathetic activation, and it also plays a role in linking emotions with memories, which is kind of that linking conscious and unconscious role of the limbic system. The hippocampus, sitting within that dentate gyrus, is important for formation and retrieval of long-term memories. So not your short-term memories, and we're going to talk about that at the end of chapter 16, memory formation, but think of your hippocampus as your long-term memory storage banks, right? And then we have a tract. Remember, tracts are white matter, so this is called the fornix. The fornix, you can see, connects to parts of the hippocampus, parts of the amygdala, and it's looping around like this, looping around into different regions of the hypothalamus. So we have the mammillary body, we have several of the hypothalamic nuclei. So all of those pieces of the hypothalamus is linked to your memories and is linked to your amygdaloid body, which is regulating your emotional states. So it's all tied in and connected together. So that is your limbic system cerebral components. And in the diencephalon, we have our anterior group of the thalamus, the hypothalamus, and the mammillary body. And it's not shown here, but your reticular formation, which is your day-night cycles, can also be linked into your limbic system, part of that, regulating your subconscious control of things, emotional states, and your fight or flight. So as you can see, it's not a region you can point to and say, hey, there's a limbic system. It's part and pieces of your cerebrum and your diencephalon. Okay? All right, so now we're on to the biggest and the last, save the best for last, cerebrum. Let me close this a little bit right. There we go. So we can see our wrinkly brains. And you might have known this is what cerebrum look like, or brains look like, is the wrinkles. And what's kind of cool about the wrinkles is they're functional. It's just not a random occurrence because of wrinkles are cool. Is the wrinkles give us increased surface area for our cerebral cortex. And cerebral cortex contains all the cell bodies, which is your gray matter. So the more wrinkles you have, the more neurons you have, the more neurons you have, the more you can process. The more you can process, the usually more intelligent you are. So the most intelligent organisms have the wrinkly brains, and that includes primates and dolphins and some other organisms. But I think those are the two most wrinkliest brains out there, dolphins and primates like humans and apes and chimpanzees. So superficially, this is a preserved human brain, so it's not the pretty color-coded one, but we can see some of these important features. So to identify, here's our cerebellum, medulla, pons. That little bump is starting to come out here. So this is all the medulla, cerebellum. Here we can see our lateral sulcus. That is separating our temporal lobe from our frontal lobe. Okay. We have what's called the central sulcus, separates our frontal lobe from our parietal lobe. And then there's a fissure back here that we can't really see. It's not shown well in this picture, and that separates our occipital lobe. Right, so we have some color-coded pictures that we're going to see as well, but those are the key features of the cerebrum in all of its beautiful wrinkliness. Okay, and there's our MRI picture showing it's the bulk of your brain mass that takes up your skull. There's our color-coded picture, so we can see those regions a little bit easier now. So frontal lobe, parietal lobe, occipital lobe, and temporal lobe. And if you'll notice, they're named for the bones that sit over them. So that should be a good review of your boneless stuff. A couple other things are highlighted here. We can see our lateral sulcus. That parieto-occipital sulcus would kind of be right here. It's not in this picture. But we have this really important one here called the central sulcus. It's like right here. It separates what's called the precentral gyrus, the wrinkle that sits in front of the central sulcus, from our postcentral gyrus, which is the wrinkle that sits just behind the central sulcus. Those two bands of neurons within that cerebral cortex are very important in sensory and motor processing and control. If we take a slice through this, kind of showing those color-coded areas still with the temporal lobe kind of on the back side there, we can see everything in relation to everything else. So this is a really good picture, or something like this nice sagittal section or lateral view for you to start putting everything together. So again, I'll just do this slowly. If you want to pause and see if you can identify things from what we've covered so far, I'll just kind of do what you can do as you're learning. So medulla, pons, cerebellum, midbrain, diencephalon or thalamus, hypothalamus, pituitary gland, pineal gland, mammillary body, right? So I got the big ones first, and you'll notice I'm going to start doing some more details. I see my fourth ventricle. I see my corpora quadrigemina. I see my cerebral aqueduct. I can see the fornix. I can see the corpus callosum. I can see the anterior commissure, posterior commissure. This is our infundibulum, pituitary gland, optic chiasm, tuberal area, cerebral peduncles. My cerebellar peduncles will be like right here, arbor vitae, right? So I could go on and on, but that's the kind of practice that you should be doing, is start with the big things: pons, cerebellum, corpus callosum. Maybe it's just those three things. And then you can say, okay, so I got those three things. Then I'm going to add medulla, cerebral peduncles, corpora quadrigemina. Learn those things. Then you get them all again. And then you just keep adding and adding and adding. So that's a kind of practice you can do when you're learning and memorizing all the parts and pieces of your brain anatomy, and do it over and over and over again. It will be really helpful for, say, quizzes, and lab practicums. Okay. All right, so we have identified all of the things we can on here. So the next thing we're going to take a look at are the white matter of your cerebrum. So again, remember, white matter are the collections of myelinated axons. There are some different collections of fibers, basically the collections of myelinated axons that have different names. So the first one are called association fibers. So the association fibers, there's two types. We have short little arcuates that kind of connect gyrus to gyrus. And then we have the longer longitudinal fasciculi that can connect lobe to lobe, say like frontal lobe to occipital lobe, frontal lobe to temporal lobe, parietal lobe to temporal lobe, parietal lobe to occipital lobe. Those are what we call the longitudinal fasciculi, and arcuate fibers are what we call association fibers because they're making associations typically on the same hemisphere. So you're not going to get a longitudinal fasciculi across opposite hemisphere, nor are you going to do that with the arcuate fibers. Then we have what are called commissural fibers. These are the ones that do go back and forth between your left and right hemispheres. The major one is that corpus callosum, and then a smaller ones we have the anterior commissure. There's also a posterior commissure that's not shown in this picture, but it would be back here. Okay, and then our last group of fibers are what's called the projection fibers. So these are all the white matter tracts that are going from your thalamus up to your cerebral cortex and back. Remember that executive secretary job of the thalamus, relaying information? All of that information is going to be traveling through those projection fibers. Okay. So now we're going to take a look at the gray matter of the cerebrum. So remember way back in our substantia nigra, way back in our brain stem, and I said we're going to be talking about basal nuclei because that is regulated by just substantia nigra. Well, we're at that point, so go back and review that if you needed to, or just open your book back to the brainstem area to find that substantia nigra. Basal nuclei, sometimes called basal ganglia, are little islands, if you will, of gray matter. So the bulk of the mass of your cerebrum is white matter. All of those tracts we just went over. So the longitudinal fasciculi, the arcuate fibers, the projection fibers, the commissural fibers, that's the bulk of your cerebrum. With the wrinkles, the cerebral cortex all around the very outside edge. But embedded deep in the middle of all that white matter are basal nuclei. So here's one view of them, kind of showing from the outside kind of this, again, that ram's horn curve pattern that we saw with our corpus callosum and the fornix and the lateral ventricles. These nuclei, some of them follow that same pattern. So here's a different view, kind of a transverse section looking down. So all of this gray matter, that's most of the processing is happening of those different regions of the brain, which we're going to get to next in the cerebral cortex areas. But deep inside, right, so we're deep inside all this white matter, we have three major basal nuclei. We have the caudate nucleus right here. We have the putamen right here. And then we have, where's the other one? The globus pallidus is not on here. Okay, let's go to the next picture. So this shows that transverse view of the caudate nucleus and the putamen. There we go. There's the putamen and the globus pallidus. Together is what's called the lentiform nucleus. So they are on kind of more of the lateral outside edge of those projection fibers, which is called the internal capsule. There we go. Those projection fibers coming out from the thalamus. And then we have the caudate nucleus. That's the ram's horn one that kind of follows the path of those lateral ventricles. Here's some of our commissural fibers. So there's the corpus callosum, our anterior commissure. Here's our projection fibers called the internal capsule. And then deep and embedded within and around all of this white matter are the basal nuclei, right? So we have our putamen, globus pallidus, caudate nucleus. So what is their job? Well, collectively together and in working with the substantia nigra, the basal nuclei work to help with coordinating your skeletal muscle movements to keep them rhythmic. They don't initiate that. That signal comes from your cerebral cortex. But once the signal to move your skeletal muscles has been initiated, the basal nuclei help to maintain the smoothness of that movement and any kind of rhythm. So think walking, right? So you consciously think, I am going to walk. It is then the basal nuclei that keeps your arms swinging and your legs moving and your muscles kind of contracting in a smooth rhythm pattern. So sometimes to help you understand how something works is to describe when it doesn't work. The disease Parkinson's disease is a problem with that relationship between the substantia nigra and the basal nuclei, that controlling regulation relationship. Dopamine is a neurotransmitter that's involved, is not functioning properly. So think of somebody who has Parkinson's disease that has a hard time having smooth muscle contraction. They have the shakes and the shimmers, and it's hard for them to do any fine motor movement. That is what happens when your basal nuclei and substantia nigra are not working properly. So when they are working properly, again, you don't even think about it because it's not, even though it is in your cerebrum, it's not part of your conscious awareness of what's happening. It kind of is this low-level background control of your skeletal muscle movements. Okay, so those are our basal nuclei. So now we get to take a look at the fun stuff of the cerebral cortex, all the stuff that we are aware of. How do we know what we know and what we can control? So we had all those lobes, right? So the frontal lobe, the parietal lobe, the temporal lobe, the occipital lobe. Within these, we have what are called primary cortexes. These are the areas of where that sensory or motor information originates or is destined. So I'm going to go through these major cortexes. So the dark green right here is your visual cortex. So your visual information goes all the way to the very back of your occipital lobe. Then we have our auditory cortex, is this yellow area. Auditory cortex is in your temporal lobe. We have gustatory, which is taste, is on the temporal and what's called the insula, which is a little fold inside your temporal. And then we have olfactory, which is smell, is also on your temporal. So we have auditory, gustation, and olfactory all located on your temporal lobe. We have vision on your occipital lobe. And then we have primary sensory right here, that post-central gyrus. Okay, so that's all your senses coming in. So visual, general sensation, hearing, taste, and smell, all on these temporal lobes. So that's all your sensory cortexes. And then in your frontal lobe, on that precentral gyrus, we have what's called the primary motor cortex. So primary motor cortex this is where your skeletal muscle signals to start. That higher level order neurons that trigger the signal all the way down to your spinal cord to tell your muscle to contract, those signals start there. So those are called our primary cortexes, so primary sensory or primary motor. So then we have association areas. So those are kind of the lighter colored regions around those darker colored regions. So for example, the visual association area right here is a lighter green. So I'm going to give you an example of kind of how this works. So I'm going to make some squiggles. I usually write this up on the board, but let me squiggle some squiggles up here. Okay, so those are some dark blue squiggles that I've put on the screen. The information that goes to your visual cortex is that these are blue squiggles. It's blue, they're linear, and that's the information that goes to your visual cortex. It has no meaning in the visual cortex. But then that information gets shared by those association fibers, those are Q8 fibers, to your visual association area, which then says, hey, based on learning, kindergarten through, your educational system, I know that this squiggle means the letter C. And I know that this squiggle means the letter A, and this squiggle means the letter R. And I also know that that means something. It's a word. So those are your association areas. They give meaning to the raw data that's coming into those cortexes. So you might hear sound, but the auditory association area gives meaning to that sound. You might feel a sensation, but then your somatic sensory association area gives you meaning to that sensation. Is a bug crawling on my hand? Is it a hair? Is it a cat tail? Whatever that sensation is, you're going to give meaning to that. So these are your association areas. They give meaning to that raw data that's coming in. That makes a little bit more sense with the sensory. For the motor, we have the motor association area over here. It's called the premotor cortex. This is taking information, kind of determining to get the information sent to start your skeletal muscle movement. Okay? And then last, we have the frontal lobe, which is not a sensory or motor cortex. That's something different. So let's continue with this analogy of my squiggles here, right? So our visual cortex said blue squiggles. Our association area said letters and maybe a word. Our general interpretive area says that word is car. And with that general interpretive area, there's links to your prefrontal cortex. There's links to your hypothalamus. There's links to your limbic system that might give you some knowledge about what a car does. So you drive a car, it might have a smell associated with it. If you've been in a car accident, you might have some bad emotional memories associated with the word car or the thought of a car. So your integrative centers help bring in information from multiple association areas to give more meaning and memory and depth and understanding to that bit of information, either a visual, an auditory, a taste, a smell, a general sensation, like a touch. All of that gets put together by these integrative centers. So that's a major job of your general interpretive area. Your frontal eye fields helps you, it takes in visual information and motor control. So you can see it's right on that boundary between the premotor cortex and the primary motor center. So frontal eye field allows your eye to scan, and we learn, as we learn English, we scan from left to right. So that allows us to continue our reading processing. The speech center, sorry, that's supposed to be Broca's. It's somehow got hooked with the prefrontal cortex. So Broca's area, or the speech center, again, right next to your motor control, that helps to control all of the musculature to have you speak, to make the movements of your tongue and your lips and your teeth and the air that's coming through your diaphragm and your vocal cords to give you the ability to have speech. So if for whatever reason you get a blockage or a death, like a viral infection or a stroke, if you damage your frontal eye field, you can't scan and read. You might read one word at a time, but you cannot scan a line of text. If you have damage in your speech center, you can't speak. You cannot coordinate the muscles and the breath needed to form words and language. If you get damage in your general interpretive area, you would be able to, and nothing else, you could see stuff. You could kind of have a meaning to stuff, but it wouldn't be connected to anything else. It wouldn't be connected to memory. It wouldn't be connected to emotions. So that would be damage there. Your prefrontal cortex, this is where your conscious, well, all of this is conscious, but this is where your kind of personality resides, your logic, your predictions, your personality. I think I already said personality. Let me find my description here. Prefrontal cortex performs abstract intellectual functions, predicting the consequences of events or actions, and has extensive connections with other areas of your cerebral cortex. Okay, so that is our cerebrum, all of the different association areas, how they take in data, how they process that data, and it's all connected together. And there's still a lot of mystery with how all of this works. Okay. The last piece for this mini lecture is hemispheric lateralization. So even though the two sides of our brain look like they are the same, they do have different functions. So for example, that general interpretive area that we just covered is only on one side of the brain. It is not on both sides, and it's typically on the left hemisphere. But if it's on the right, it's not going to be on the left. The other thing that we didn't see in that last picture is this area here that is responsible for spatial visualization and analysis, and you only have one of those, and it is on the opposite side of your general interpretive area. So if you are have your general interpretive area in your left hemisphere, your spatial analysis will be on the right hemisphere, or vice versa. So your vision comes in, is split between your two eyeballs to your two visual cortexes. We're going to see that in our visual pathways in chapter 17. So hemispheric lateralization is just the idea that each hemisphere is similar but different. And your left or your right hemisphere controls the left side of your body and receives information from your left side of your body. Your left hemisphere controls and receives information from your right side of your body. So there's a crossing over in that information that goes up and comes from your hemispheres. Okay, so that is all of our brain structure. The last video that I'm going to put for chapter 14, I'm going to talk about the brain activity, the brain waves, and then a short little review of your cranial nerves. So that's something you'll be working on for a lot in lab with your memorizing of your cranial nerves. Okay, so I'll see you then. Bye.